All right, chapter 20, fluid and electrolyte balance, and we'll also look at pH balance. So this goes in conjunction with the kidneys. One of the concepts we didn't go over at the beginning of the semester is about mass balance. There's a little bit about it in chapters 1 through 4, but basically we have a balance of water and a balance of ions and electrolytes in our bodies. And we have systems in place to regulate that balance. So if we get too much, then there's a system in place to excrete excess, and then if we don't have enough in our bodies, then there has to be a way to introduce it back into the body. All right, the primary route for getting rid of excess ions and excess water is going to be the kidneys in the urinary system, because a lot of water is lost in the filtrate that is gathered to make urine. But we can also lose some ions in our feces and then also as we sweat. And then the lungs, when we breathe in and breathe out, whenever we breathe out, there are still some water droplets in our breaths. We lose some water through the lungs as well. And then whenever we change carbon dioxide into bicarbonate, water is used to make carbonic acid. So how do we add more if we're deficient? If our bodies are low in water, if we get dehydrated, then we have the thirst mechanism, which is housed in the hypothalamus. Makes you thirsty, so you will intake and drink more water in order to make you more hydrated. And then if we have a low amount of electrolytes like sodium and chloride, then we have cravings for salt. That's why we crave potato chips and all that good stuff. When it comes to the systems that will help regulate your fluid and electrolyte balance, they can use the respiratory system and the cardiovascular system, and they are faster. They create a rapid response compared to the other system, which is going to be the kidneys. It's a lot slower to put things into and out of the filtrate that eventually become part of your urine. Let's look at these mechanisms. What happens whenever there is a decrease in blood pressure or an increase in blood pressure? What are the coping mechanisms? So if we look on the left, if there is a decrease in blood volume, then there's also going to be a decrease in blood pressure. And that can be a problem, because if there's a decrease in blood pressure, then there's not going to be enough of a pressure gradient to get nutrients like oxygen and glucose to the tissues where they need to be and to get rid of the wastes. If there's a decrease in blood pressure, then there's not going to be enough going through the kidneys that you can excrete through the nephron. All right, so if there's a lower blood volume, which creates a lower blood pressure, that is detected by both the atria and also the baroreceptors in the aorta. The three mechanisms, we have first the cardiovascular system. So how does the cardiovascular system compensate for a lower blood pressure or lower blood volume? It does it by vasoconstriction, by stimulating the sympathetic nervous system, which will then create vasoconstriction of those blood vessels. So if the blood vessels vasoconstrict, it narrows the lumen, increases resistance, and then that will also increase the blood pressure. Now let's look at the kidneys. How does a low blood volume and low blood pressure affect the kidneys? Well, the kidneys can conserve water, and they can conserve sodium chloride or salt, which will raise the blood pressure and raise the blood volume. And then the nervous system, what does the nervous system do? The central nervous system will then stimulate the thirst center, which is located in the hypothalamus, so that you intake or drink more water. Whenever you drink more water, then there will be more water introduced in the extracellular fluid, which will then go into the intracellular fluid and then increase your blood pressure. Now let's look on the right side. What happens if there's an increase in blood volume? If there's an increase in the blood volume, then there's an increase in the blood pressure. And again, there are going to be receptors that will detect this increase in blood volume and in blood pressure. To compensate, we can use the cardiovascular system and the kidneys. We'll look at the cardiovascular system first. So if there's an increase in blood volume, which increases the blood pressure, then the cardiovascular system will then vasodilate. Not vasoconstrict, it will vasodilate. And vasodilation will decrease resistance, which will then increase flow, and it will lower the blood pressure by vasodilation. Now let's look at the kidneys. If there's an increase in blood volume and an increase in blood pressure, the kidneys will then excrete excess sodium and excrete water into the urine. Once it's in the bladder, it's not considered part of your internal environment, so it lowers your extracellular fluid and your intracellular fluid volume, which will then lower your blood pressure. This picture will explain mass balance. We'll look at the left side of the picture. Water gain. How do we get water into the body? We ingest 2.2 liters per day just in the food that we eat and the things that we drink. So we intake 2.2 liters that way, and then through metabolism, using aerobic respiration, it also produces some water, so that the total for the whole day intake of water is 2.5 liters per day. So how do we get rid of excess water? First, we lose 0.9 liters a day in what we call insensible water loss. It's through the skin and through the lungs. We're not aware of it. As we breathe out, our breath has some fluid in it. Our skin will sweat, and then it'll evaporate the water. And then we excrete 1.5 liters per day in the urinary system, and then we get rid of 0.1 liters per day just with the feces. And that adds up to 2.5 liters per day. So if we take in 2.5 liters a day and we get rid of 2.5 liters per day, then overall we haven't changed the water level in our bodies. So that is mass balance. In the last chapter, we talked about the GFR, which is the glomerular filtration rate. How much is filtered through the renal corpuscle in a minute or so? And in this picture, they show you with the glomerular filtration rate. As long as the water levels are above a certain amount, which I'm circling right here, then we have water that's going through the kidney nephron, and then we reclaim most of that back, meanwhile losing just a little bit out as part of the urine. In this picture, it shows you, though, if our water levels go below a certain amount, that means the blood pressure, hydrostatic pressure, is too low, and there's not going to be any more fluid leaving through the nephron through the filtration membrane. Let's highlight some important information on this slide. The title of it says the renal medulla creates concentrated urine. So let's review. What is the renal medulla? The renal medulla is the renal pyramid that's in the middle of the kidneys. And the renal pyramids are full of collecting ducts, plus some of those nephron loops from the juxtamedullary nephrons. So the renal medulla creates concentrated urine. When we looked at the chapter on urine formation, the last chapter on the kidneys, we had filtration, then we had reabsorption, and then we had secretion. And then whenever the urine is in the collecting duct, the osmolarity of the urine in the collecting duct varied somewhere between 50 milliosmoles up to 1200 milliosmoles. And we're going to look at some pictures, and I want us to get some concepts down before we start looking at some of the pictures. One of the things to remember is that the medulla, the tissues, has a high osmolarity, especially as we get closer to the renal papilla. So that means that the fluid that's in the descending loop of Henle is going to lose water to the medulla, and then that water that's lost into the medulla will then be picked up by the peritubular and the vasorectal capillaries. And then the cells in the ascending limb of the nephron loop, they're impenetrable to water. There are no water channels in the ascending limb of the nephron loop, but they are actively transporting sodium out of the lumen and into the medulla. So instead of losing fluid, it's losing solute. And if you lose solute, that means it becomes more diluted. So that means the fluid that, like I just said, that's leaving the loop is more dilute than the fluid that entered it. If we just remember from back in chapter 19, that the osmolarity of the filtrate as it leaves the glomerular capsule is 300 milliosmoles. But then by the time it goes through the nephron loop, it's then going to be 100 milliosmoles because it lost fluid and then it lost ions. And then the distal collecting duct, which is part of the distal convoluted tubule, which is part of the distal nephron, it usually has no water channels in it, but it does become permeable to water. It becomes permeable to water because of a hormone known as vasopressin, also known as antidiuretic hormone. The antidiuretic hormone will open water channels to allow more water to filter out of the lumen and then into the tissues so that they can be returned and brought into the circulatory system. All right, let's follow the light in this and see what's happening. First, we have this line right here. This line separates the cortex of the kidney from the medulla. So here's the cortex up here, and here's the medulla down here. The osmolarity of the tissues of the cortex are 300 milliosmoles. The osmolarity of the medulla starts out at 300 milliosmoles, the part that's closest to the cortex, but then as we go down deeper into the medulla, osmolarity increases. So we go from 300 to 600 to 900 to 1200 milliosmoles. The numbers aren't that important, just the concept. We start out at 300 milliosmoles, so the number gets higher. What does that mean if the osmolarity gets higher? That means it becomes more concentrated, which means the osmotic pressure is going to be higher if you want to look at it that way. So we'll keep that in mind as we are looking at the filtrate as it goes through the distal and the proximal convoluted tubule and as it goes through the nephron loop. All right, so as the filtrate enters into the proximal tubule, it has the same osmolarity, so it's isosmotic to the tissues that are surrounding it. It's isosmotic to the cortex, and then as it goes into the medulla, it's also isosmotic to the medulla in the start at the top of it. So it's at 300 milliosmoles. What then happens when we get into the descending limb of the nephron loop? It tells you right here, only water is absorbed on the descending limb of the nephron loop. So if we lose water and we don't lose ions, what does that do to the osmolarity of the filtrate? It's going to increase the osmolarity because we're losing solvent, we're losing water. So we're losing water to the tissues. So what happens to this 300? Now it's going to get higher and higher because we're losing water and we're retaining solid ions. So that when we get to the very bottom of the loop of Henle, the osmolarity is 1200 milliosmoles, which is the same osmolarity at the deepest part of the renal papilla or the renal medulla. So the osmolarities match, they match, they match, they match because we're losing water from the nephron loop. It's going into the tissues, but then it goes into the capillary, so it's not staying in the tissues. And then let's see what happens when we go through the ascending limb. At the ascending limb, there are no water channels, so the water cannot cross through the membrane. It stays inside of the renal tubule. However, there are ion gates, and ions are going to leave the filtrate. So if we take a solution and we remove ions but keep the water, then the osmolarity is going to get lower because it's becoming more dilute. I wish I could write this out on the board for you. So more diluted means less concentrated. Why is it less concentrated? Because we're retaining water and we're losing solid particles. We're losing ions to the tissues that are surrounding. So the osmolarity that started at 1200, once we start losing ions, the osmolarity is going to go down, 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 down. And then by the time it exits the nephron loop and goes into the distal convoluted tubule, it has an osmolarity of 100 milliosmoles. All right, so let's take a break and look at this. If the filtrate in the distal convoluted tubule is only 100 milliosmoles, what's the osmolarity of the tissues surrounding it? The osmolarity of the tissues surrounding it it's 300 milliosmoles. So 300 milliosmoles means the tissues of the cortex have a higher osmolarity than the filtrate inside of the tubule. So if there were plenty of open water channels in the distal convoluted tubule, where is water going to go? Water is going to go from the tubule into the cortex. Why? Because water goes to a higher osmolarity. It's going to go from 100 milliosmoles towards the 300 milliosmoles to dilute it. However, normally, without any hormones present, then there are very few, if not any, water channels, aquaporins, in the distal convoluted tubule. Same thing with the collecting duct. So if there are no water channels, then the osmolarity that we started at right here at 100 will still be around 100 when it gets to the very end of the collecting duct. But let's say there is a hormone present. Let's say there is antidiuretic hormone and these water channels have opened up, so now water is going to go to the cortex. So now the osmolarity will get higher because we're losing water and we're not losing ions. If we keep losing water as we go through the collecting duct, then the osmolarity will get higher. So it could be possible for the osmolarity to be around 1200 milliosmoles, the same osmolarity at the very bottom of the renal medulla. So vasopressin, also known as antidiuretic hormone, it comes from the posterior pituitary. So when vasopressin is released, it is released constantly, but as if the vasopressin amounts increase, then the amount of water channels increases. That's what they mean down here by a graded effect. There's a direct relationship between the amount of vasopressin released and the number of water channels. And the more water channels that are present in the distal convoluted tubule in the collecting duct, the more water you're going to retain, because that's what vasopressin, antidiuretic hormone, is going to do, make you retain more water. Now, why would you want to retain water? Usually it's because you have a lower than normal blood pressure or lower than normal blood volume. So vasopressin acts on the collecting ducts and also on the distal convoluted tubule. It stimulates aquaporin insertion, so it opens up aquaporin channels to allow water to leave the filtrate and go into the tissues and then be collected by the vasorecta or the peritubular capillaries. I would like to point out that vasopressin is secreted in a certain pattern during your circadian rhythm. As we sleep during the night, then less vasopressin is released, so that less urine is produced at night than during the day. So let's see what's happening through these pictures. On the picture on the left, we have maximum amount of vasopressin being released. The picture on the right, letter B, there is no vasopressin being released. All right, so if the maximum amount of vasopressin, antidiuretic hormones, being released, we're going to lose water. So the osmolarity, as we lose water, will get higher and higher. So we start with an osmolarity of 100 milliosmoles, and then as it loses water, the osmolarity goes up. As the water leaves the collecting duct, it goes into the vasorecta. So the osmolarity keeps getting higher and higher and higher. Now let's look at the osmolarity of the blood inside of the capillaries. The osmolarity of the blood capillaries starts around 1200 milliosmoles, but then as it collects the water, the osmolarity is going to get lower and lower because it's becoming more dilute, until it's around its normal, around 300 milliosmoles. Now if you look at the right-hand side, no vasopressin, no antidiuretic hormone, then whatever the osmolarity of the urine is when it goes into the collecting duct is basically going to be the same osmolarity it's going to be as it makes its way to the bladder. There are three main triggers of release of vasopressin. First, there's a decrease in the blood pressure. Second, there's a decrease in atrial stretch because of low blood volume. Or three, the third trigger, osmolarity that is greater than 280 milliosmoles. So osmolarity, remember, when it's higher, that means there's not enough water in the cardiovascular system. So we have to retain more water. So let's start with the decrease in your blood pressure. What does that do? It's detected by the carotid and the aortic baroreceptors, which then create a stimulus that is picked up by the hypothalamus, and then hypothalamus triggers the release of vasopressin. Vasopressin makes you retain more water, and then that will increase your blood volume and your blood pressure. The decrease in atrial stretch because of low blood volume, that is detected by the atrial stretch receptors, the proprioceptors of the atrial muscle tissue, which then is detected by the hypothalamus, which will then trigger the release of vasopressin, which will then open up the aquaporins in the collecting duct, which makes you retain more water, and then your blood pressure, blood volume goes back up. Osmolarity. If an osmolarity of greater than 280 milliosmoles, that means that your blood is pretty thick. That is picked up by the hypothalamus, the osmoreceptors that can tell the stretch in cells. Once the osmoreceptors of the hypothalamus have been triggered, then it triggers the release of antidiuretic hormone vasopressin, which will then open up the channels in the collecting duct, which will make you retain more water. And then as you retain more water into your system, then the osmolarity of your blood will go back down. This picture is showing you that there's a direct proportional relationship between the amount of vasopressin released compared to the plasma osmolarity. So as the plasma osmolarity increases, then the amount of vasopressin being released increases until the osmolarity will come back down to normal. All right, anything that is a counter current multiplier, that means we're going to have basically two types of tubes carrying fluids going in opposite directions. We have a counter current multiplier when it comes to the pampiniform plexus that surrounds the blood going into the testes and the scrotum. The pampiniform plexus surrounds the spermatic cord. We went over this in anatomy. It surrounds the spermatic cord so that the testicular blood or the blood in the testicular artery going towards the testes, the heat can be transferred to the series of veins that surround it, so that the blood entering into the testes is about 3 degrees cooler than body temperature, because that's the maximum temperature allowed to make the most sperm cells is about 3 degrees cooler than body temperature, which is why the testes are outside the body and not inside of the body. But that's just an anatomy review, and basically I'm talking about the counter current multiplier, because we have the same thing in the kidneys. We have two fluids going in different directions. We have the filtrate going one direction towards the renal papilla, and then we have the blood, which is exiting the nephron going towards the renal vein. All right, so here are the two currents. We have the filtrate going in one direction, if you follow the red dot, and we have blood going in the opposite direction, if you follow that red dot. We follow the filtrate first on the left-hand side. As the filtrate goes to the descending limb of the loop of Henle, it's losing water. That's what they're showing here. We're losing water, and it's going into the vasa recta. So as we lose water, the osmolarity gets higher, which is why we go from 300 milliosmoles to about 1200 milliosmoles. And then when we get to the ascending limb, the water stays where it is, but now we're losing ions. So the water stays inside of the lumen. We're losing ions. So now the osmolarity gets lower. So the osmolarity as it exits through the ascending limb is about 100 milliosmoles. Now what happens to the water and the ions that we lose? Some of the ions that are lost in the ascending limb stay in the renal pyramid, which is why it has such a high osmolarity. But other ions are going to enter into the vasa recta. So if we look at the osmolarity of the blood entering the vasa recta, we start at 300 milliosmoles, which is about normal. We start gaining the ions. So as we gain ions, the osmolarity gets higher. It goes from 300 to 500. And so now when we get to the bottom of the loop around the vasa recta, it has an osmolarity around 1200 milliosmoles. Well then, as we go through the rest of the vasa recta, it's going to collect the water coming from the descending limb of the nephron loop, plus the water that is lost by other parts of the vasa recta. So now the osmolarity gets lower because we are gaining water, so that the osmolarity of the blood as it leaves the vasa recta is the same osmolarity of the blood that went into the vasa recta, and that's the countercurrent multiplier. We are going to be gaining ions one side and then gaining water on the other side of the cardiovascular system, the vasa recta. And then in the nephron loop, we are just losing water in the descending limb, and then we're just losing ions in the ascending limb. All right, we talk about sodium balance for a second. There are two different hormones that will increase or decrease your blood sodium levels. The hormone that increases your blood sodium levels is aldosterone. Aldosterone comes from the adrenal cortex, and when it's released, it opens up sodium channels so that you can increase the amount of sodium that's being collected from the filtrate in the urine. So aldosterone will increase your blood sodium levels. Then we have atrial natriuretic peptide, that is ANP, that comes from the heart muscle of the atria. When it's released, it does the opposite of aldosterone. It will lower your blood sodium levels by making more sodium stay inside of the filtrate, so it's not reclaimed by the kidneys. All right, so if we look at sodium chloride balance, wherever sodium goes, chloride follows. If we have excess sodium chloride in our system, then it will raise the osmolarity of the blood. And as we looked at earlier in a couple slides, an increase in osmolarity triggers the release of vasopressin. And vasopressin will then make you conserve more water, which will then lower your osmolarity. In the nephrons and the tubules, the reabsorption of sodium is an exchange gate. It exchanges sodium and potassium. So aldosterone will not only increase your blood sodium levels, it decreases your blood potassium levels. So you trade one for the other. Another trigger for releasing aldosterone is low blood pressure. When your blood pressure gets too low, then the adrenal cortex will release aldosterone in order for you to retain more sodium, and whenever you retain more sodium, you also retain more water. I think this tried to be a little redundant. When you ingest excess salt, and if there's no change in volume, then it increases the osmolarity of the blood, which is then detected by the hypothalamus. Hypothalamus then triggers the release of vasopressin. Vasopressin increases the amount of water that is retained from the kidneys. As the kidneys conserve water, then the osmolarity will be lowered. The other thing that will be triggered is your thirst mechanism. So when you increase your salt intake, which will decrease your osmolarity, it increases the thirst. So as you get thirsty, you drink more water. When you drink more water, then you increase the extracellular volume and you increase the blood pressure. When that happens, then the osmolarity will return to normal and your blood volume and blood pressure then returns to normal. The three triggers for the release of aldosterone is if there's been a decrease in blood pressure, if there is very high osmolarity, or if there is an overabundance of potassium. Remember, potassium is the ion that the extracellular levels will affect your resting membrane potential. So if your potassium levels go high, then aldosterone is triggered to be released. Now, if there's a decrease in blood pressure, that triggers something called the RAS pathway, the renin-angiotensin system pathway. And there's going to be more on that later. But all of those triggers will then stimulate the adrenal cortex to release aldosterone, and then aldosterone will increase sodium reabsorption. At the same time, it stimulates potassium secretion. And remember, secretion is whenever an ion goes from the internal environment to the external environment. So secretion usually boils down to two different ions, either potassium or the hydrogen ion. All right, here's the renin-angiotensin pathway, or the renin-angiotensin system. There are cells called juxtaglomerular cells. These juxtaglomerular cells are located in between the afferent and the efferent arterioles, plus also that part of the distal convoluted tubule that passes right by where the junction of the that place where the afferent arterioles are located. These juxtaglomerular cells secrete renin, and renin is a part of the renin-angiotensin pathway. Renin is an enzyme that converts something called angiotensin to angiotensin 1. Angiotensinogen is an inactive protein enzyme that is located in the blood, and it is made by the liver. So renin converts angiotensinogen, which is an inactive zymogen, an inactive protein enzyme, to angiotensin 1. And then there is an enzyme called ACE, ACE, angiotensin converting enzyme. They used to think that ACE only came from the lungs, and there are some tissues in the lungs that produce this enzyme. But come to find out, angiotensin converting enzyme is also made by the endothelium of your blood vessels. Now this enzyme, it converts angiotensin 1 to angiotensin 2, and there are different effects of angiotensin 2, and we're going to see it on the next slide. So here are the functions of angiotensin 2. Angiotensin 2 increases the secretion of vasopressin. Vasopressin is the antidiuretic hormone, and then the antidiuretic hormone will then stimulate you to reabsorb more water from the filtrate because it opens up the water pores, so it increases your blood pressure or increases your blood volume. So angiotensin 2 stimulates the release of antidiuretic hormone. Angiotensin 2 also stimulates thirst. So if your thirst has been stimulated, then you drink more water, which will increase your extracellular fluid volume, increases your intracellular fluid volume, and then increases your blood volume and your blood pressure. Angiotensin 2 is also a vasoconstrictor. So it makes your blood vessels constrict, which will raise your blood pressure. Angiotensin 2 also stimulates the sympathetic nervous system output to the heart. So the sympathetic nervous system increases your heart rate. So an increased heart rate increases cardiac output, which will then increase your blood pressure. And then angiotensin 2 also increases reabsorption of sodium. So it retains more sodium from the filtrate. It increases how much sodium is reabsorbed by increasing your aldosterone levels. There are some medications called ACE inhibitors. So if we are taking an ACE inhibitor, that means we're going to be inhibiting the enzyme that converts angiotensin 1 to angiotensin 2. So there are many blood pressure medications for people who have high blood pressure that are called ACE inhibitors. The ACE inhibitors will then inhibit the production of angiotensin 2, which has all of these effects that will raise your blood pressure. So it helps suppress angiotensin 2. So like I mentioned earlier, the opposite of aldosterone is going to be ANP, atrial natriuretic peptide. It comes from the tissues of the heart, from the atria. They will make you get rid of sodium through your urine, so it increases the amount of sodium that's in the urine. And it does that so that it will decrease your blood volume, which will decrease your blood pressure. So if your blood pressure gets high or blood volume gets high, then excess sodium will be lost through the urine. Wherever sodium goes, water follows, so then it lowers your blood volume. I'm not going to mention much about potassium balance. We went over potassium a little bit in the nervous system in chapter 8. So potassium and sodium have like an opposite relationship. If we retain more sodium from the filtrate, then you excrete or secrete more potassium into the filtrate. And then to do the opposite. If we get rid of excess sodium into the filtrate, then we reabsorb and take in more potassium from the filtrate. And the reason why potassium balance is so important is because of hypokalemia and hyperkalemia, because it affects your resting membrane potential. If you don't have enough potassium in your blood, that's hypokalemia, and it causes muscle weakness and the failure of the diaphragm to contract, and then it makes the heart sluggish. With hyperkalemia, when you have an excess of potassium in the blood, it leads to an excess of, or an increase in your heart rate, which can cause cardiac arrhythmias. Now, what can cause disturbances in your potassium levels? Well, people with kidney disease, they have trouble processing potassium. Excessive diarrhea makes you get rid of excess potassium through your feces, and then using diuretics. I'm going to go ahead and skip the next few slides because that's what I would have done in lecture anyway, and just go ahead and go to acid-base balance. What is pH? pH is the measure of your hydrogen ion concentration. So normal pH of blood is between 7.38 and 7.42. Normally it's around 7.4. That is the pH that is used by most of your enzymes to be activated and to work. So if we stray too far off of this pH balance, then it affects your enzymes, and it affects your breathing, it affects your contractions of your muscles. Plus also changing the pH can also denature your proteins. And when you change the conformation of the proteins, that's what denaturing means, then it changes their function and they're no longer able to work properly. All right, whenever there's a change in your pH, we have acidosis and alkalosis. Acidosis means that you have a higher than normal concentration of hydrogen in your system, so you're acidotic. And when that happens, then your neurons are not able to be excited as easily, and it suppresses or depresses the central nervous system, makes you sluggish. And then in alkalosis, that means that your hydrogen ion concentration is too low, your pH is getting too high, then the neurons become hyperexcitable. And then if that happens, then it leads to muscle tetanus because they are receiving potentials at a faster rate than normal. So what can cause some of these pH imbalances? Well, if we take in too many fatty acids or amino acids in our diet, or if we increase the amount of lactic acid or keto acids, like whenever you just eat a protein diet, then that's going to increase your hydrogen ion input. And whenever you increase your hydrogen ion input, then it's going to lower your pH and make it too low. It's going to go lower than 7.4. So how do we compensate for that? We compensate by excreting or getting rid of excess hydrogen ion or using buffers. We have buffers in extracellular fluid, bicarbonate. The proteins and the hemoglobin, phosphate in cells, they are alkaline, so they can help buffer some of the acidity of the increase in hydrogen ion coming in. And then we have the ammonia and phosphates that have a negative charge to also help. But then we also use the respiratory system and the renal, the kidneys. So through breathing in carbon dioxide, if we can breathe off excess carbon dioxide, then that will change your pH. Or the kidneys can either secrete or reabsorb excess hydrogen ion, and that also will balance out your pH. Let's review a couple of terms: hypoventilation and hyperventilation. With hypoventilation, that means you're taking shallow breaths. When you take shallow breaths, your oxygen levels will decrease and your carbon dioxide levels will increase. If your carbon dioxide levels increase, then it's converted to carbonic acid and it's going to lower your pH, which means it's going to make you more acidic. Hyperventilation is whenever you're breathing really rapidly and your oxygen levels will go up, but your carbon dioxide levels, you're going to be blowing off more carbon dioxide, so your carbon dioxide levels will lower. If your carbon dioxide levels lower, that makes you more alkaline. It's going to raise your pH. Let's look at the next picture. This picture is showing the respiratory compensation for metabolic acidosis. Well, first of all, what is metabolic acidosis? Acidosis means that you have a higher than normal hydrogen ion concentration, which means your pH is too low. If it's a metabolic acidosis, then it's something that's going on with metabolism, and usually it has something to do with the kidneys. So the kidneys are not getting rid of excess hydrogen ion like they should. So with metabolic acidosis, that means you have an increase in your plasma hydrogen levels, which means you have a decrease in your pH. That is the amount of hydrogen ions detected by the chemoreceptors in the aorta and the carotid. So then it stimulates the respiratory center in the medulla, which will increase the diaphragm contractions, which means we're increasing the rate and depth of breathing, and if that means we're hyperventilating. So when we hyperventilate, it lowers your plasma concentrations of carbon dioxide, and as we lose carbon dioxide from the system, then your hydrogen ion levels will drop and your pH will go back up. So that will feed back up here and then shut that off. All right, now let's see what happens over here on the right-hand side. If we have an increase in the plasma hydrogen ion concentration, which means there's a decrease in the pH, then that also means that there's an increase in the plasma amounts of, or plasma partial pressure of carbon dioxide. So these are the two triggers for hyperventilation. If there's a higher than normal amount of hydrogen ion, which means your pH is too low, or if there's a higher than normal amount of carbon dioxide in the plasma. Both of those are going to be detected and increase your breathing rate. If you increase your breathing rate, it decreases your carbon dioxide levels, which will then lower your plasma lower your plasma hydrogen ion concentrations, which means it raises your pH. All right, so what is the renal compensation for acidosis? Well, what the kidney is going to do if your pH is too low, if your hydrogen ion concentration is too high, then it will try to secrete more hydrogen ion into the filtrate, into the tubule. And remember, this is the external environment, and anything that's in the external environment is not considered a part of the body. So as soon as that hydrogen goes into the external environment, then it'll help raise your pH. Let's go over this real quick. Respiratory acidosis means that your acidosis has to do with the respiratory system. So that means your pH is too low. That's what acidosis means, which means your hydrogen ion levels are too high. Respiratory acidosis is caused by hypoventilation. They're the same thing. So respiratory acidosis is caused by hypoventilation, which means you're not breathing enough oxygen in and you're not breathing enough carbon dioxide out. So that leads to an increase in the partial pressure of carbon dioxide in your system, which means your pH is going to decrease. So respiratory acidosis is a hypoventilation problem, which means your levels of carbon dioxide are increasing, and the levels of carbon dioxide and your pH have an inverse relationship. As the carbon dioxide levels increase, the pH of your system decreases because you're becoming acidotic. Now let's look at metabolic acidosis. Metabolic acidosis means you're acidotic. That means you have a lower than normal amount of hydrogen ion concentration and your pH is low. Usually that's due to your dietary and metabolic input of hydrogen ion exceeds excretion. So that means you're increasing the amount of hydrogen you're putting in your system through your diet and more than what your kidneys are excreting. And when that happens, then your pH is going to decrease because you're becoming more acidotic. Remember, the lower the pH, the more acidic you are. The higher the pH, the more alkaline you are. And then we have respiratory alkalosis. That means that you have a lower than normal amount of hydrogen ion, which means your pH is too high. It has to do with the respiratory system. So respiratory alkalosis is due to hyperventilation. That means you're breathing in a whole bunch of oxygen and you are breathing out a whole bunch of carbon dioxide. If you're breathing out a whole bunch of carbon dioxide, then that's going to lower your carbon dioxide levels. So your carbon dioxide levels go down, and when your carbon dioxide levels go down, your pH goes up and makes you more alkaline. So that's respiratory alkalosis. All right, then we have metabolic alkalosis. So with metabolic alkalosis, we have a loss of hydrogen ion, usually due through excessive vomiting or excessive ingestion of bicarbonate-containing antacids. Overdosing on your Tums or Rolaids will cause a metabolic alkalosis. So you have a loss of hydrogen ion through excessive vomiting or an increase in taking too many bicarbonates. And when that happens, that's going to increase your pH and make you more alkaline. So it's a metabolic alkalosis. All right, so that is the end of this chapter for me. So the next chapter will be the digestion chapter. Email me if you have any questions. This can be a little confusing when we start talking about increasing and decreasing hydrogen ions, which means you increase or decrease the pH. Remember, as you increase your hydrogen ion concentration, it decreases the pH because you become more acidotic. But if you get rid of excess hydrogen ion, then it's going to raise your pH, which means you're becoming more alkaline.